thank you for letting me talk about some of the quantitative phenotype data curation we're doing, and specifically in the HRDP strains, because we've heard a lot about this, this panel in previous talks. So the hybrid rat diversity panel, just as a reminder, is a panel of 96 different strains that were selected to have genetic diversity and are really helpful for genetic studies, especially for complex traits. So the goal of the MCW Hybrid Rat Diversity Program, which is an R24 funded to Dr. Dwinell, really is to acquire and rederive the strains that are in the Hybrid Rat Diversity Panel, to maintain these and distribute these strains to the scientific community. They're also doing whole genome whole genome sequencing using Illumina short reads. Again, that data will be shared to the scientific community through the public archives, but we're also collaborating with RGD for data deposition. So RGD has developed a landing page, if you will, for the HRDP strains, and this will be centralized and created a centralized resource in order to have phenotypes and genotypic data in this centralized place. So I'm going to talk about the strains and our work annotating their associated phenotypes, and Monica Tutai will be talking about the genetic variation in the HRDP strains next. So for instance, if you click on the WKY in this classic inbred strain from the landing page, you'll be taken to the strain page at RGD for that particular substrain. And you can see the different information that's available, including its origin. And if we know any source data, that's something we're going to have to keep working on to keep updating the source data. But RGD is often, for years, has been doing phenotype curation, mainly for genes, QTLs, and strains. But the main area of curation has been using the mammalian phenotype. And the disadvantage of that is that it is based on abnormal phenotypes. And so some time ago, Mary Shimayama had started this phenominer project to be able to annotate quantitative data from the strains. And in order to do that, you really have to gather some additional information. And so this includes the actual trait, the measurement that is being reported, the method of how that measurement is being made, and any external experimental conditions, including tissue sources or cell sources or other types of experimental conditions. So if we go back to the WKY, you can see that if you on this right-hand side in the summary, you can see the various annotations that have been made. Some of these are disease annotations, such as in the WKY, you could go in and see hypertension. Well, that might be misleading until you go down to the mammalian phenotype and it actually says decreased systemic arterial blood pressure. And then you can actually go down into the quantitative measures so that if you click on systolic blood pressure, it will collate different studies that have measured blood pressure in the WKY. So you can compare and contrast different conditions where you may find increased or decreased blood pressure in that particular strain. So when we went to the hybrid rat diversity panel, we realized that there were several of these strains that had no annotation. So we decided that that was a good goal to do. So we started annotating these 8 strains that didn't have any quantitative data. And the workflow for that is to go to PubMed and start pulling any papers that mention those strains. You have to go through triage to make sure it actually has quantitative data that you can extract from the paper. And then that paper is annotated to bring in the measurements and the values of that particular measurement being done, along with the other terms that go along with that particular study. Now, so far, the group has annotated 128 papers, added almost 3,500 annotations, quantitative annotations for those 8 different strains. And at the same time, in order to be efficient, those same papers are curated for their mammalian phenotypes if there is a comparison strain to identify differences or abnormal phenotypes. And again, those are annotated just as well. So in all, we have curated literature with annotations for disease and phenotype, both qualitative and quantitative, totaling nearly 9,500 annotations for the HRDP strains, along with variants for 48 HRDP strains compared to both RN6 and the new BN rat assembly, which Monica will talk about next. I just can't have to call out that there are other people working very, very hard on multiple data types and resources for the HRDP, including the Hybrid Rat Diversity Program I mentioned, as well as Phenogen, the folks in Prague in Japan who developed the recombinant inbred strains, and the scores of researchers who have developed and who are studying the HRDP strains in their independent research. So with that, I thank you.